Can you hear me okay? Yes. Good. All right. Thank you for coming. This is another one in our series for the School of Neuroscience. This is one of our faculty candidates that it's my pleasure to introduce. This is Dr. Masashi Tabuchi, who is currently a postdoctoral fellow in Mark Wu's lab at Johns Hopkins University. And Masashi, he got his PhD at the prestigious Graduate School of Engineering at the University of Tokyo. And he did his work in the Kanzaki lab studying functional organization of the olfactory system in silk moths, looking at pheromone regulation of behavior. And he's actually the first person ever to use optogenetic techniques and whole cell patch clamp recording in this species. So it's really pioneering work. His work resulted in 7 publications, 4 first author publications, and he actually graduated in 3 years with a PhD. So it's pretty extraordinary. Yeah. And so after graduating, he then joined the Wu lab to work on temporal coding and how that underlies circadian rhythm behavior in Drosophila. So, and he continued to innovate with optogenetic and electrophysiological recording techniques, doing some more pioneering work using those techniques. But also he's added to his repertoire a series of molecular techniques to further expand on how he can address this issue. And actually, looking over his application, a comment from his current mentor, Mark Wu, really spoke to kind of put it into context. So Wu said that Masashi's work is truly seminal, and he said, quote, "I believe this study represents the most important work my lab has ever done." Pretty high praise. Masashi, he's actually incredibly productive. He's published over 14 articles in many prestigious journals, including Neuron, Science, PNAS, and Cell, including a publication, another one coming out in Cell. That'll be 2. He has numerous awards, and there's actually too many to list, but of course one very important award is that he currently has a K99 through the National Institute of Neurological Disorders and Stroke. And it's also important to note that he has significant mentorship experience where he's currently supervising, or he has supervised, 5 students in just the Wu lab alone. So it's my pleasure to introduce to you Dr. Tabuchi, and the title of his talk today is Patterns Matter, Temporal Codes in Neural Signaling, Synaptic Plasticity, and Behavior. Turn it over to you. Oh. Video. Roxanne can help. Technical difficulties. There we go. Leave it to the experts.
Okay.
Can you turn on your microphone?
Yeah.
Okay.
Yeah, so thank you so much for such a great introduction. First, I'd like to thank the search committee for inviting me to speak today. Also, yesterday, actually, I had a great time to speak with a faculty member yesterday, although flight was a little bit delayed. So today, I'd like to share my recent work, including how spiking pattern of circadian neuron regulates sleep quality. So basically, from my PhD student period, I have a long-lasting interest in how a neuron encodes information. And particularly, as a neural code, I have interest in studying the non-canonical form of neural code that I have to explain later. So yeah, that's it. So maybe I want to get started. So this is kind of an introduction slide. So as I said, I'm interested in how neuron encodes information. So as you can see, these are animal So, most of animal mass process information, including external environment or even internal state, is themselves. So this is an example of kind of information processing of external environment. So as you can see, this bear now try to capture the fish, but now obviously fish is moving fast. So that's why this bear mass process kind of both visual processing or even kind of sensory integration system in his brain. So that's why kind of high amount of high computational processing required in his brain. So this is kind of example of how neuron processing for the external object. Then this is kind of another example of the kind of internal state of brain. So as you can see, this lion now try to kind of drink the water, despite having a zebra near his side. So this is kind of clear example that how animal behavior is depending on the internal state of brain. So now probably I guess maybe the lion now kind of feels super thirsty, so that's why kind of he really prioritizes drinking water compared to kind of capture zebra. So so this is an example of kind of sensory processing, and this is an example of internal state processing. And during my PhD, when I was a PhD student, I focused on kind of sensory processing aspect. And in my postdoc period, actually, I focused on internal state. So, well, at the biophysical level of neural code, this is also kind of a very introduction slide. But information neural coding should be happened within the brain network. And as probably everyone knows, the information is processed by kind of dendrite as input and the axon as output, and they can communicate each other via the synapse. And as a signal, they tend to generate action potential, which is spike, as electrical activity of neurons. So this is kind of fundamental unit of neural code. So everyone probably tend to think, as saying here, kind of action potential could be a communicative language for neuron as neural code. However, as a broad scheme, spike is not only the way to process the information. So as you can see, neural code, neural code should be classified into spiking-based information processing and even non-spiking-based processing. And I will explain later, but I think probably most of people really focus on kind of rate coding, which is basically quantified by mean firing rate of spiking cell. However, as I said, my interest is more kind of a non-canonical form of neural coding. So maybe rate coding can classify it as a canonical form of neural coding. Then, as I circled here, this kind of thing, processing can be classified as a non-canonical form of neural coding. And yeah, I will explain later, but temporal coding basically, spiking pattern or spiking timing are used to encode information. And this is more complicated, but as I said, neuron does not have to use action potential to perform information processing. Rather than they just can use just a fluctuation of subthreshold membrane potential as analog coding. So because I said analog because maybe spiking can be digital code. So that's why for example, even non-spiking neurons, such as some types of retinal circuits, are actually using analog code to encode the visual information. And moreover, even in the central nervous system, some neurons, including several types of neocortical circuit or even hippocampal neuron, are using analog code besides the spiking code. So that's why my today's topic is focusing on the non-canonical form of neural coding. And this is kind of one slide showing my PhD work. So when I was a PhD student, actually, I was trying to study the CPT problem. So CPT is kind of chemical plume tracking. So that means how we can detect and track the plume, chemical plume in the air to approach the origin of odor source. And as a model system to study CPT, I use the silk moss as a system. So this is because actually silk moss has an amazing, outstanding olfactory system in terms of sex pheromone detection. So this is female and this is male, and as shown in this blue plot, female moths are releasing a sex pheromone called bombicol. And by using this sex pheromone, male moths can approach the female. So during this pheromone guided orientation processes, male moths should address at least two difficult problems, which is basically kind of specificity and sensitivity. So in terms of specificity, sex pheromone molecule is not only chemical in the air, because as you can see, there's maybe some plant odorant or some non-related chemical components should be also existing in the air, but male moths must just specify the pheromone molecule, and moreover, they also detect. They also need to detect kind of several molecules in the air. So that means this system also requires ultra high sensitivity. So I was fascinated by these questions. That means how male moths can detect and specify the sex pheromone to approach the female. So basically, as you can see, this female moths exhibit kind of mating dance behavior, which is a kind of zigzagging and a kind of surging, looping kind of programmed behavior in response to detecting the sex pheromone in the antenna. And as you can see, this is schematics. This is first order olfactory circuit in the moth, which is called antenna lobe. And basically, sex pheromone molecule is detected by the olfactory receptor neuron. And this information is processed in the synapse. And the postsynaptic neuron, called the projection neuron, convey the information to generate this mating dance behavior. And my main finding during my PhD work is that there is unusual temporal integration in the synapse, which happened within an 80 millisecond interval integration time. And this was functionally used to integrate non-simultaneous olfactory input to amplify the spike output to effectively generate behavior. And the meaning of 80 milliseconds is, surprisingly, this 80 milliseconds are matching the time scale of chemical plume dynamics in the air. So that means this is already optimized to maximize the information and amplification of this signal to effectively convert the behavioral output. So this is one evidence that temporal coding as a spike integration was functionally used to generate behavior. And after my PhD, I decided to study Drosophila as a model to understanding the internal state of the brain, as I mentioned early slide. Then basically, as shown here, what I mainly found is that the spiking pattern, but not firing rate in the clock neuron was regulating sleep quality under the control of clock gene. That is kind of main discovery and actually take-home message of this presentation. So because, as you can see, this process was initiated by circadian clock. So that's why let me briefly explain kind of circadian clock gene network. So this is basically kind of circadian clock molecular oscillator. So as a Nobel Prize was awarded last year to Dr. Hall, Rospash, Yang for understanding this molecular machinery of circadian oscillator, which is basically, there are two transcriptional activator drive the expression of transcriptional repressor to form the transcriptional translational negative feedback loop. So this is the machinery of circadian clock oscillator. So now it looks like this molecular core mechanism itself is well established, well understood. However, we still don't fully understand how this clock translates the timing information to regulate the downstream output, including feeding, or body temperature, blood pressure, or even sleep. So in this aspect of circadian regulation of sleep, actually our lab previously identified a novel circadian output molecule called wide awake. Our lab found this molecule is critical for regulating clock-dependent sleep onset. I will explain about this molecule briefly later slide, because I use this molecule a lot as control the circadian output. So here, let me briefly explain about fly sleep, because probably everyone think, what is fly sleep? So basically, I want to say fly sleep was already established since 20 years ago, actually. Then basically, fly sleep was quantified by consolidated inactivity, just like how Fitbit or Apple Watch measure your sleep, but based on kind of consolidated inactivity, right? So as you can see, fly sleep clear all aspects of human sleep, including as I said, consolidated immobility, or even they have arousal threshold, and they also have a homeostatic aspect in response to sleep deprivation. And they are also controlled by circadian clock, as I said, and even monoaminergic dopamine function. And this is interesting, but even fly is very sensitive to caffeine to decrease sleep. Fly sleep can be measured by locomotor activity, and this is very high throughput. We can simultaneously record 32 fly sleep. Then we can trace the sleep profile as just like this plot. So then, in terms of sleep aspect, probably most of sleep researchers focusing on the effect of loss of sleep or sleep amount effect. But I am more interested in sleep quality, because as shown here, it has been shown that the sleep quality is also important for regulation of our performance during wakefulness, or even our emotional stability. So that's why we should have we always should have a very good sleep quality during our sleep. So then I want to talk about my data to record sleep and sleep quality in fly. So this is kind of raw data of control fly sleep over 24 hours. This is kind of Zeitgeber time, which is basically the time after light onset. And this is just fly sleep amount based on 30 minutes beam size. And this axis is also kind of sleep quality measurement, which is basically based on a brief awakening number during sleep. And as shown here in these blue traces, basically nighttime actually fly sleep as napping, and during daytime fly sleep more. And in terms of sleep quality, as you can see in this red trace, their sleep was highly fragmented during sleep, which is basically same as our napping. We cannot nap 6 hours, 7 hours. On the other hand, during nighttime, there is significant decrease in brief awakening. That means sleep quality is good that means kind of sleep was more consolidated, which is basically the same as our sleep. So, that means there is a difference in terms of sleep quality during daytime and nighttime. And as shown here, ZT6 to 8, ZT 18 to 20, I particularly focus on these two time windows to isolate the effect of sleep amount. This is because, if we compare these two time windows, as quantified here, actually there's no difference in terms of sleep amount. However, there is significant difference in terms of sleep quality. So I just thought this is a very good, nice opportunity to isolate sleep quality effect from the effect of sleep amount. And because I was thinking, actually, maybe this sleep quality difference between day versus night was depending on clock gene. And yes, as you can see, actually, the sleep quality was depending on the clock gene and even clock output molecule wide awake dependence. So yeah, as quantified here, this trace, the difference in terms of sleep quality between day versus night was not observed in clock mutant and wide awake mutant. So this really suggests that the sleep quality between day versus night was really depending on circadian clock gene and even a clock output molecule wide awake. So then, next question, I naturally wanted to ask how this happened. Then I was thinking maybe because clock gene was expressing in circadian clock neuron, so that's why maybe some firing activity is regulating sleep quality. So to explain this, let me first briefly explain the neural circuit of circadian clock neuron in fly. So basically, in the clock neuron, a clock circuit in mammal, I think just a suprachiasmatic nucleus is the only termination. However, in fly, they have more detailed classified in terms of cell type identity. So as you can see, the clock network was initiated by a large NV, which is a kind of master pacemaking neuron, can act also as a photorecipient. And this information is propagated through the small NV to the DN1 cell. And the DN1 neuron is the final output at the clock circuit. And this information is transformed to non-clock circuit, which is basically a PI. The PI has arousal effect. And in this topic, I particularly focus on DN1 cell because DN1 has been shown in regulating the sleep arousal modulation in fly. Moreover, as you can see, there is a monosynaptic connection between DN1 and PI, so which makes it easy to analyze the effect of circadian clock circuit to non-circadian clock arousal promoting circuit. So yeah, this is the result of recording from large DN1 cell, which is shown here. So I just focus on two time windows that I measure sleep quality. And this is a daytime firing pattern, and this is a nighttime firing pattern. So first, I want to emphasize that there is no difference in terms of mean firing rate, which is good because we can exclude the contribution rate coding. On the other hand, as you can see, there is a significant difference in terms of spiking pattern between day versus night. I will explain later, but briefly, spiking pattern is very kind of irregular in nighttime, but during daytime, spiking pattern become more kind of regular, which means kind of precisely timed manner in terms of spontaneous action potential firing. So there is clearly difference. However, similar to sleep quality, this kind of difference in terms of spiking pattern was not observed in clock mutant and wide-awake mutant. So that means this firing pattern difference between daytime, nighttime also depending on clock gene and wide-awake gene. So in other words, this firing pattern difference between day versus night are actually correlating with the sleep quality difference between these time windows. So that's why I might be able to say, actually, these are driving the sleep quality. But as I said, this is correlation, but we always have to show the causation in addition to correlation. So that's why I really want to test by using causation effects by using optogenetics. However, yeah.
So those are recordings from the DN1 neuron.
Yes.
Did you look at any other neuronal populations in that circuit to see if they exhibit similar properties in firing patterns?
Yeah, actually, I did because the reviewer asked. So basically, I only yeah. In addition to DN1 cell, in response to reviewer's request, I also record from large NV, and essentially, it has a kind of same phenomena. Although precisely it's different, actually. I have to spend more time to explain what is the difference between this and this in terms of spiking pattern. But actually, essentially it's same, but slightly different. Yeah.
While we're on that, I ask you a quick question. If you go back,
Yeah.
you have at a minimum 2 cell types within DN1,
Yeah.
the ones that are expressing wake and the ones that are not. Do they show a difference in their pattern at this the daytime sleeping?
Oh yeah, actually that's a good question. So I had a technical difficulty in terms of optogenetics, but at least in this experiment, we actually record only from DN1 expressing wake.
Okay.
Oh yeah, that is actually very important point to qualify. Yeah. As you can see, not all DN1 express wake, so that's why we also generated a driver to label only wake positive cell. So that is what we mainly used. Although we had to use some different driver which may contain a wake negative cell, but in other experiments, but at least. In this experiment, I just record from the cell expressing wake. Yeah.
For your recording and for those dark times, do you do them in light? How do you control their still dark time?
So basically, I basically kind of wrap red film. So basically, that should be kind of invisible for fly visual system. So basically, as I mentioned, I try to, of course, we had to use light, but I kind of cut all kind of blue light, green light, except for red light for my visualization. So so that's why I think the effect of light should be minimized. Yeah. So let me take a step back to explain rate coding, temporal coding again, because this is important. So, as you may know, rate coding is the idea that a spiking frequency encoding the information. So that means more spike or less spike can differ. Then on the other hand, temporal coding is the idea that the spiking timing or pattern are encoding the meaningful information to generate behavior. There is several types of neural coding are well known, including fast spiking latency code, or even STDP, spike timing dependent processing, or even at the population level for neural dynamics, they can always also act as macroscopic dynamics as oscillation or even kind of synchronization activity. But in my case, I more focus on pattern, particularly intrinsic pattern, which can be generated by spontaneous action potential train. And I also mentioned that particularly intrinsic pattern as really hard to study because it's always coexisting with rate coding. So that's why we really have to try to exclude the contribution of rate coding to assess the sort of function of pattern coding. And moreover, I also wanted to emphasize that molecular mechanisms underlying intrinsic pattern are largely unknown. So that's why, based on this question, I wanted to ask how this can contribute to the information processing to generate sleep quality difference. And for the technical aspect, intrinsic pattern as temporal code can be quantified by coefficient of variation, which is basically just can be calculated by interspike interval variance. And probably this, as you know, but if CV shows high, more than 1, that means the spiking pattern can be irregular pattern. On the other hand, if CV value shows very low value, that means this is a kind of regular pattern, which is precisely timed action potential sequence. So I decided to synthesize the CV 1.2 pattern as a daytime code and the CV 0.3 as a nighttime code. This is because when we always introduce these patterns as optogenetic stimulation, we really need to exclude any contribution of late coding. So we try to just use raw data as spike pattern generation, but raw data always has some variance in terms of rate coding, firing rate difference. So that's why we really had to synthesize this pattern by just utilizing mathematical modeling. So we did. So basically, we tried to perform machine learning to capture the feature of this pattern between daytime and nighttime. And basically, as you can see, we could kind of recapturate the experimental feature to the model. Both, so this is kind of low-dimensional description, which is basically based on the probability first-order probability density of function. But moreover, we also could recapturate the characteristics by showing the joint distribution of interspike interval. So that means actually, as you can see, they could contain second-order structure in terms of temporal sequence, which is shown this kind of feature. So basically, as shown here, we describe the pattern by using a modular approach to use it as an optogenetic stimulus pattern. Then, by using this code, we try to test these two pattern difference in actual behavior. So this is a fly, and fly was tailored. Then the fly movement was monitored by IR camera, and DN1 neuron now expressing optogenetic molecule, which is CS crimson, which can be activated by red light, but not blue light in channel option. So then, in response, under this condition, we simulate this LED by using that synthesized pattern I showed in previous slide. So this is a kind of really pure examination for understanding the effect of a pattern without changing mean firing rate. Yeah.
For the CV.3, when you do the autocore, do you see a clear theta or whatever frequency modulation, or have you done the smooth power spectrum? What do you see the
I don't know. I have, but I have autocorrelation data. I hope I have. Maybe I don't have. I hope I have. Maybe not. I'm sorry. I don't have. But yeah, they show very beautiful autocorrelation. That autocorrelation data actually will be shown in my paper, which will be published actually this Thursday. So if you want to,
So I'm just curious, what is it at? What is the is it at theta frequency? Is it 4 hertz, 5 hertz? What do you see?
Oh, basically about 3.3 hertz. Yeah. And as I said, it's really showing very beautiful autocorrelation. That means the pattern is really.
Peak every 300 hertz in your autocore.
Yeah.
300 milliseconds, okay.
Oh, yeah. Yeah, so sorry, actually, I should put autocorrelation. Yeah, anyway. So basically, as I mentioned, at least, yeah, we could perform testing of different spiking pattern under the identical mean firing rate. Then we are hypothesizing maybe irregular firing can trigger fragmented sleep and regular pattern induce consolidated sleep. And yeah, this is frame subtraction captured by this camera. As you can see, the leg movement was detected by optic flow. I mean, this is very basic paradigm, but so basically, by detecting optic flow subtraction, we created a sleep profile shown here. Yeah, so basically, blue is detected as sleep, and red is detected as brief awakening, and green is detected as awakening. So under the stimulation of CV 1.2, as you can see, compared to CV 0.3, the sleep was highly fragmented. And CV 0.1 sleep was highly consolidated, which is quantified by bout duration of the sleep. So longer bout duration means more consolidated sleep. So this clearly suggests that temporal spiking pattern alone can drive the sleep quality behavior with clear causation. So I think this is pretty good as evidence of contribution of spiking pattern without using mean firing rate as rate code.
So can I?
Yeah.
So when you, in the other condition where it's irregular spiking mode, when you have an ISI of about in the same as when you have the CV.3, does that actually induce quiescence in the?
Yeah.
Those periods where it's irregular, but if you have the, approximately in the 3.3 hertz firing, does that those irregular periods also induce, or is there something else going on?
Actually, yeah, exactly. It also could induce irregular, fragmented sleep. Yeah.
I was wondering whether the spiking alone with that particular statistic does it, or you need some other sort of neuromodulatory.
Yeah, I think in this case, maybe based on this, because if we slightly change the spiking pattern within the range of CV 1.2, still we could induce sleep fragmentation. So that means maybe such precise pattern may not be required. Yeah. However, it's not published, but actually, I think we discussed yesterday, but actually we also tested unidimensional coding. That means, because as I said, the spiking pattern of daytime code has some second order structure based on, temporal structure, just shown here. So if we can lose this, but we can keep this shape, but we can lose this. So in this case, actually, sleep fragmentation was slightly rescued. So that means actually this might be contributing a lot. Yeah. So, and as I mentioned briefly, another question that I want to tackle by using power of genetics in fly is, molecular mechanisms underlying the temporal code. And as you can see, I performed the genetic screening by using a CCAP neuron, which is, there is CCAP neuron here, and this CCAP neuron is regulating wing expansion during eclosion. And because the wing expansion is depending on the excitability of a CCAP neuron, first, I found that if we express wake, excitability of a CCAP neuron was decreased, and the decreased excitability of CCAP neuron actually triggered unexpanded wing. And actually, we are thinking, oh, this is a good indicator for finding the genetic interaction of wake by using RNAi approach. And as you can see here, if we also introduce RNAi knockdown of gene of interest, we can rescue the wing expansion, unexpanded wing, maybe throughout disturbing the molecular pathway downstream the wake signaling. So, and based on this system, we perform large-scale screening, more than 1,000 of genes. Then we kind of identify the top hit molecule showing a high rescue value. So let me briefly explain what are these molecules. Basically, these are kind of the same, which is kind of a slow. Slow is calcium-activated potassium channel, and SLOB is its binding protein. So that means they are kind of functional coupling. And NAK beta is ATPase beta servant, which is completely different. I will discuss the molecular function of this for spiking pattern later. But so in terms of SLOB, kind of this is KCA channel. Slow molecule, which is calcium-activated potassium channel. This is structure, and slow is known to bind calcium near the calcium ball to alter the conformational structure to alter the inactivation state of this KCA current. So that means slow has functional coupling with KCA. And this is a direct measurement of KCA current from DN1 cell. And interestingly, DN1 current also has circadian-dependent difference in terms of their amplitude. So actually, this is actually similar with the finding in SCN.
Just one other, do you know if the time constant of KCA is around that of the 3.3 hertz?
So in terms of rise time, I think it shows 61 millisecond or something like that.
So it's not setting that ISI for those spikes.
Yes, it is. But I think I can discuss this aspect later.
Okay.
Yeah, it's slightly different. Yeah, but I would say maybe in this paradigm actually another molecule may be contributing. So yeah. So as you can see, there is a difference in terms of peak conductance. And this conductance was not observed in wake mutant or even a slow knockdown with gene-specific manner in DN1 cell. And with correlation of this, the after hyperpolarization of action potential of DN1 cell are also showed similar tendency, which is not surprising because they should be kind of have functional coupling. Then that's kind of a difference was not observed in any clock mutant, wake mutant, or even slope knockdown. And these difference of spike waveform are actually correlate with spiking pattern. It's shown here. So this is also not surprising because HP contributions for spiking pattern are well known. But as you can see, the difference in terms of spiking pattern between day versus night was not observed in slope knockdown, quantified here. And with correlation of this result, the sleep quality was also changed. Nighttime sleep quality was also changed after introducing slope knockdown. So that means slope are responsible for changing these spiking pattern or even sleep quality at the behavior level. And as I shown in previous slide, besides slope molecule, I was also finding the NAK beta subunit as a second hit molecule. And I was trying to study the functional significance of this. So this is just a schematics of NAK ATPase, and beta subunit is known to regulate alpha subunit current activity just by binding this kind of shape. And basically, because they are regulating the balance of between extracellular or intracellular concentration of sodium distribution. So that means, in other words, this is regulating the cooperativity of voltage-gated sodium channel because cooperativity of activation of sodium channel is depending on the partially depending on the distribution balance of sodium ion itself. So this is really complicated, but I think this may be relating what we talked. So this is not spike, actually. This is not spike. This is first derivative of spike waveform, which can be quantified as the indicator of spike onset rapidness. So as you can see, there is clear difference of spike onset rapidness between day versus night. So during daytime, spike onset rapidness has more variance in terms of time to peak. And the time to peak itself also is not high. However, compared to daytime, nighttime has more coherent time to peak. And the time to peak itself is very rapid. And as shown here, these differences were not observed in clock mutant, wake mutant, or even NRK knockdown. So that really suggests this difference of spike onset rapidness also depending on clock gene pathway. Yeah, quantified here as a first derivative parameter. So together with Rob, I'd like to explain how this exactly causing spiking pattern sleep quality. Well, actually, I forgot things. So similar to Slope, yeah, the knockdown of NRK beta also affecting spiking pattern and sleep quality. So that means these two molecules are kind of similar function in terms of spiking pattern alternation or sleep quality alternation. But we think, yeah, their output as a spiking pattern change, sleep quality change are kind of the same, but I want to argue they are contributing different aspect for biophysical parameter, although the output is same. So to explain that aspect, let me basically summarize. So my finding so far. So as you can see, wake regulating two molecules that I identified by large-scale screening, and these two molecules are contributing, kind of spike onset rapidness and after hyperpolarization. Then these two change of biophysical parameter of single action potential can cause the change of macroscopic action potential pattern. Then this can increase the sleep quality. So this is what's going on under the wake control. So yeah, I just wanted to address the question, how these two molecules are regulating this biophysical parameter. So this is actually a simulation of Hodgkin and Huxley model. And this is random noise that we used for generate spontaneous action potential because unlike pacemaking cell in natural state, model cell does not have spontaneous activity. So that's why we always had to induce a random current to activate neuron. Then what is remarkable feature of this is, as you can see, when we injected kind of depolarizing current, as you can see, addition of KCA are affecting as brake. However, if we introduce NRK molecule when the current was hyperactivated, hyperpolarized, compared to control, actually they are speeding up. They are making a speed up their spiking pattern. So then this is kind of reverse experiment, but this is experiment. So this is simulation. That in this simulation and these experiments, we kind of also knock down slope and NRK beta as shown in previous slide. Then the interspike histogram also showing a kind of opposite phase of this, which makes sense because this is knocked down, but this is knocked in. So that means in the absence of slope, ISI distribution are kind of shifting to the left side, which means because they are kind of acting as brake. On the other hand, if we knock down NRK, they are introducing the ISI introducing a kind of so this means kind of speed up. So because this is knockdown effect, then if we add NRK beta, they act as a brake. Oh, sorry, this is complicated
I
actually.
just want to make sure I understand.
Sorry, yeah, this is very complicated, so
So
hard to explain.
In a lot of contexts, when you
Yeah.
have something like KCA, which hyperpolarizes it,
Yeah, yeah.
it engages currents like IH or IT, and
Uh-huh.
especially IH, which imposes this kind of theta
Yeah,
time scale.
Yeah.
Here, you added additional, on top of the KCA, you added sodium channels. What have you added in line 3? I don't follow what you did in line. What is line 3 showing?
so in this line 3, these are all independent experiments. So yeah, so this is a mimic as daytime. That means there's no KCA conductance and there's no NAK current. And this I only add KCA parameter.
Sure, I got that one, but the line 3,
Line 3 I only add NAK.
but you kept the KCA.
No KCA. Yeah, there's no KCA in this data.
Okay.
And in fourth traces, I implemented both KCA and NAK. Yeah. So, yeah, so it's kind of complicated, but so this is gain of function, but this is loss of function. Yeah. But basically, I wanted to say, these two molecules are contributing different aspects of, yeah.
I just want to make sure I understand this part. But for the NAK, what does that exactly mean for the what conductances did you say change? You removed the KCA. You just you increased NA conductance, or what does NA/K mean in this context?
So in this Hodgkin and Huxley simulator environment, this basically can be referred as just pump current conductance.
You increased the pump conductance.
Yeah.
But that, okay.
Yeah, because probably pump current may be regulating indirectly for the cooperativity of voltage-gated sodium channel activation, but by regulating the counterbalancing the sodium ionic balance of concentration. So.
But these cells don't have any hyperpolarization activating cation channels or anything like that.
Yeah, we don't implement. Yeah.
Okay.
Yeah.
Because that's usually the mechanism that often with KCA, often you get IH. That would impose theta time scales.
I see. Yeah.
Just curious, because you're getting something around approximately low theta.
Yeah.
That's another possibility in my mind.
Yeah. The reason that I did not implement IH current is we could not find IH current in these cells.
You did not find it.
Yeah, and even another researcher also testing the RNA-seq from this DN1 cell, but they could not find the gene also. Yeah. So, and the final question, I also wanted to study how this temporal code is transformed to downstream cell to generate behavior. And I hope you remember this brain image. Because I was recording from here, and the downstream is here, which is PI cell. And Amita Segar's lab already showing PI cell has a rate coding change between these time windows. So that's why, based on this, actually, I also could reproduce her result. But basically, based on this, I was thinking, there is rate coding difference between these time windows. So that's why maybe temporal coding of DN1 is transformed to rate coding in the downstream cell. So to test this, I perform intracellular recording from PI neuron, then perform optogenetic manipulation again in DN1 to manipulate the spiking pattern. Yeah, then as I mentioned in previous slide, I use these two stimulus patterns, then just observe what happened. Then, so this is the result of showing firing rate change during optogenetic stimulation. And as you can see in this blue trace is, a green trace, maybe first, let me explain green trace. So, green trace is the PI neuronal response during CV 0.3 optogenetic activation. Then as you can see, basically nothing happens. However, if we introduce an irregular pattern, which shows the red trace here, so during first 5 minutes, nothing happened, but they suddenly increase the firing rate. And this increased firing rate isn't even kind of sustained even after the simulation time. So that means this is kind of could be a plastic change to keep the persistent activity of a PI neuron. And importantly, this was also not observed in the presence of NMDA receptor knockdown in this neuron. So that means maybe postsynaptic NMDA receptor may be involved in this phenomenon. And I just analyzed the same data set, but focusing on EPSP. And as you can see, EPSP also showing some change in terms of both slope and amplitude, and correlated with increase of firing rate. EPSP amplitude and the EPSP slope increased by only irregular pattern of DN1 neuron, but not irregular pattern or even NMDA receptor knockdown in PI cell. So that means this really suggests that there is a synaptic plasticity induced by temporal coding of DN1 to induce the rate coding in PI neuron. So kind of this is a kind of summary of my recent finding. So during nighttime, wake regulating two molecule, as I mentioned, then this contributing spike pattern generation as precisely timed manner. And that has, this phenomenon kind of increases sleep quality. But as I said, during daytime, the spiking pattern exhibits a very irregular pattern. Then this irregular pattern actually inducing the potentiation of PI cell as synaptic plasticity. And this potentiation of PI neuron actually decreases the sleep quality because the PI neuron is basically arousal promoting neuron. So this is my research accomplishment so far. Then maybe by using a few slides, I also briefly mention what I want to do in the future. Yeah, and as I mentioned, I am interested in non-canonical form of neural coding. So that's why, yeah, I want to study temporal coding furthermore. And even as I mentioned in introduction slide, I also want to study analog coding in addition to temporal coding. So kind of this is my first project that I want to launch in the future. So basically, my recent research accomplishments showing what happened during nighttime mainly. But we still don't know what's going on in the daytime to induce this potentiation. But particularly, I was really curious what's the contributing factor to generate an irregular pattern, and that is kind of one sub-aim. So I think maybe there's some channel noise can contribute to this spike pattern generation, as well as extrinsic growth of neural sequence to generate complicated structure of action potential firing. Then, I also want to know how this potentiation is triggered. So I think I really want to perform genetic screening of this neuron to identify what molecule downstream of the receptor are forming this neural potentiation, which is basically induced by spiking pattern alone without firing rate changing. And the second topic is a little bit tricky. So in this DN1 PI synapse, I also found STDP besides SPDP. So I said SPDP is spike pattern dependent processing, and STDP is well known, right? Spike timing dependent processing. And I just want to emphasize timing and pattern are completely different. So because timing is basically just determined by relative timing between presynaptic action potential and postsynaptic action potential. But the pattern is determined by just high dimensional temporal structure of only presynaptic action potential. So that's why I just wanted to emphasize they are completely different. Timing and pattern are completely different. But nevertheless, I also found spike timing dependent plasticity in the same synapse, and that's even depending on the daytime and the nighttime. So basically, during daytime, STDP exhibit anti-Hebbian form, which basically induce LTD. On the other hand, during nighttime, STDP in the synapse showed Hebbian form, which is basically LTP. So that means, their output of STDP in the synapse are completely opposite during day versus night. So I really want to know what's going on in the synapse. And one of the controversial issues of STDP is what STDP doing at the level of behavior output. And it's hard to examine this, but I think this synapse might be a good opportunity to dissect this STDP function in the behavior. So I also want to do this as a second sub-aim. And as I emphasize, this synapse has both SPDP, STDP, so and pattern dependent and timing dependent. So they might functionally interact to generate multiplex code. And that should be computationally powerful. So that's why I just want to know whether these are interact with each other to forming multiplex code. And this is the final topic. So this is completely different that I was talking. So besides the circadian clock, I was also characterizing the neuron coding protein hunger. So this is completely different aspect, but I just want to use this neuron as a model to study analog coding. So I have to briefly explain what's this neuron. So with collaboration with Dr. Chiri Liu, and now she's a UCSF. So this neuron, we found that this neuron are encoding the protein hunger state in the brain. So what is protein hunger is a little bit complicated. I want to minimize explanation. So we tend to think hunger is a unitary form because based on our requirement for total calorie. But actually we found that the hunger can be classified with nutrition specific manner. So that means we have multiple hunger depending on we now need sugar, we now need protein, now we need lipids. But this neuron only can respond in response to protein hunger, which is kind of interesting. And this neuron has two branches. And this neuron are connected with sugar processing center. So that's why I call sugar branch. And this branch are connected with protein processing center, which I call protein branch. And as you can see, if we record from the axon of this branch, actually the firing rate was kind of the same. So that means these two branches showing both same firing rate. But as you can see in this subthreshold membrane fluctuation, sugar branch is really kind of silent in terms of subthreshold dynamics. On the other hand, in the protein branch, as you can see, it's fluctuating a lot. And I am hypothesizing this might be used as an analog code, as E pre-SP. Not EPSP, E pre-SP. That means because this is axon, so this is axon, so that's why it could be possible that this is excitatory presynaptic postsynaptic. Excitatory presynaptic potential as analog code. So I just want to know what's going on in this branch-specific subthreshold activity. And as you can see here, besides analog coding, it clearly has spike. So that means maybe analog code and digital code are interact synergistically as a hybrid code. So that is also kind of interesting topic, I think. So that's why I also want to study how spike and non-spiking dynamics are coordinating to generate a meaningful code, to generate protein hunger perception. So basically that's it. So finally, I'd like to thank my postdoc mentor, Mark Hu, and collaboration that they taught me kind of computational, stuff. And Grace is an undergrad in my the Mac lab, who basically contributes genetic screening. And Ben is a PhD student in our lab. Actually, his data is not shown, but we also perform immunoprecipitation to test the biochemical interaction between wake and slope. Yeah, he performs. So. And they are kind of my senior postdoc, now who are both independent in VIB and UCSF. So, and of course, I also thank the funding. Thank you very much.
Questions during the presentation, but we got time for a couple of questions.
So when you use optogenetic to control the Gen1 neurons, how do you control the intrinsic spikes?
So basically, actually, I realized that only the neuron called pacemaking cell can be controlled by optogenetics. Yeah, because as you said, actually, I also tested other types of neuron, but I could not control. So I think this is because pacemaking cell always has the activity of pacemaking cell always depending on the intrinsic spike generation ability without synaptic drive. But many other types of neuron spike are generated basically synaptic drive. So that's why, if so, how we can control by optogenetics. But what we found that if we utilize optogenetics in clock neuron, which is a pacemaking cell, actually we can really hijack the spiking activity. So yeah, I have that, I think. Yeah, so this is the verification of optogenetics. So kind of I don't know how to. Yeah. So basically, if I kind of increase the light frequency, yeah, somehow the difference will be what happen if I increase the frequency too much. Maybe this is one of the phenomena called frequency adaptation. However, as you can see, as a physiological range that this neuron intrinsic has, such as kind of maybe 3 Hz or something, at least within this range, we could capture the timing very precisely, and that is shown here. So yes, there's some mismatch firing, and of course, there is variability in terms of spike latency after light onset, but I think this is still reasonable control in terms of spiking pattern. And even we had the extra spike, but at least if we quantify the activation pattern by using CV, LV, CV2, at least the pattern itself is kind of no different. So that's why overall shape of a spiking pattern should be preserved. So yeah.
Can I ask a question about M2 that you had about the...
Oh, M2.
Yeah. So at least in this context, it seems to me that when you say pattern, you mean rhythmic, right? So if you have some sort of a regular in the input, that it's going to give some regular submembrane threshold oscillation or the ongoing oscillation, then that's going to kind of determine what phase the spike is going to come in on. So I would expect that to translate to
I see.
kind of plasticity. So is it, what's the real difference there, I guess, is that what you're trying to argue for? So that it seems like the pattern seems like it means it's rhythmically firing. Then if the output of that rhythmic firing has some sort of ongoing oscillation, then it's going to, whether it comes in at the trough or the peak, that's going to really affect whether the cell is excitable or inhibited, and that's really going to change the timing of the two spikes. Essentially, then in that regime, what you really have is just spike timing dependent plasticity. So can you just elaborate a little bit more on
Sure. Yeah. So basically, the most important point here is, yes, both pattern, timing cause plasticity, but in the same time period, their direction was opposite. So as you can see, during nighttime, as I said, during nighttime, they follow a Hebbian rule, which is basically potentiation. But during daytime, they are showing anti-Hebbian, which is basically depression direction. So if so, but at least during nighttime, spike pattern-dependent plasticity, I did not observe. I could observe spike pattern-dependent plasticity during daytime, and spiking pattern-dependent plasticity during daytime shows potentiation direction. So that's why they are kind of counterbalancing the new synaptic state by bringing the different direction of the membrane excitability. So I don't know how to say. So because maybe this as a potential function of daytime STDP, maybe this can be used as a break to prevent overloading of the potentiation that is caused by spiking pattern.
I mean, like in the hippocampus, depending on the phase that the spike comes in for the theta rhythm, so there's the REM stuff where the novel cells go into the trough, which you get LTE, and you go to the peak, you get LTP. So I'm just wondering whether it's just a matter of what's receiving it being in a slightly different state.
Yes, it's possible, but as I mentioned, because both potentiation and depression could be happening in the same synapse with same synapse. So that's why I don't have to. So I mean.
Let's talk about it offline.
Oh yeah, sure.
We talk about it offline.
Yeah.
I think we need to move on. We had a lot of questions during the presentation, so let's another round of applause. I have a question.
Oh, okay.
So.